I'm just making sure that everyone's come into the room. Yeah, thank you very much for attending this event. My name's Bridget Howarth, and I'll be chairing today. And there's time for questions at the end of the event. And so do put them in the chat as we go through, and then there'll be some facilitation of those questions at the end. So I'm Bridget Howarth. I work at the university in the Innovation Impact and Business Directorate. It's a large knowledge exchange team at the university, and a lot of our work does indeed focus on research that's linked with the culture and creative industry sector. So I'm really interested to hear what the panel have to say about this project today. And what I'll do first of all is introduce the speakers. So we have three speakers today. We have Professor Elaine Bignell, Co-Director of Research for the MRC Centre for Medical Mycology. Léonie Hampton, an artist with regional and international perspective and currently artist in residence at the Centre. And Professor Neil Gow, also of the MRC Centre and Deputy Vice-Chancellor for Research and Impact at the university. So we'll be sharing some images during the discussion, and it'll be quite interesting to see what you think about the images as well. So first of all, we have Elaine, and Elaine is going to set the scene and perhaps talk about what the mission of the centre is and what new elements are added by working with an artist. Elaine, would you like to start?
Bridget, very much. The MRC Centre for Medical Mycology was created in 2016 and it was set up to address the critical problem of fungal diseases of humans. Why do we need a centre that's focused on this problem? Well, over a quarter of the world's population suffer from fungal infections, and most of these affect the skin, the hair, or the genitourinary systems, and they're not life-threatening. But many millions of these infections can be as deadly as Ebola if not properly diagnosed or treated, and they kill more people than malaria. And we've all witnessed appalling recent examples from India of the devastation that these organisms can cause in susceptible patients. And the high rates of mortality that we see with these diseases are an indication of our current inability to effectively tackle them. So we urgently need drugs, diagnostic tools, and a better understanding of the disease processes that are involved. Next slide, please. The work of our centre is founded upon world-class research that addresses many aspects of fungal form and function, interactions of fungi with their human hosts, and the translation of this research into the know-how and drugs needed to tackle the fungal threat to life. Next slide. But this bedrock of excellent research supports three other major goals of the centre. And these are to build capacity in this field, both in clinical and in basic research, and to migrate this knowledge and capability to all corners of the globe via our international centres. The fourth and equally critical goal is to educate and engage all sectors of the population about the importance of fungi, both as life forms and as pathogens of human beings, because they are relatively new causes of infectious diseases and they are often overlooked. Next slide. An important part of our current 5-year vision is formed around a programme of engagement that was masterminded by our excellent communities engagement officer, Lorna Barnes, who may be able to join us later. But this is geared towards changing public perceptions of fungal disease, involving the public in a two-way dialogue about research, and strengthening a culture where researchers value public engagement. and also to inspire and galvanise young audiences to engage with the importance of science and the topic of medical mycology. So our work with Léonie is a new and exciting part of our engagement journey that is proving to have impacted us all already, a lot more than we initially imagined. And it's a pleasure to be here today to talk about where we're at with that journey. Thank you.
Thank you, Elaine. That was really useful. And I think, Léonie, when we've talked about the preparation for this event, it was really interesting to talk about scale and the nature of research, and to moving into an area about which we know very little sometimes. So do you want to talk a bit more about that?
You're mute, Léonie.
Thank you. Absolutely, yeah. Thank you. It's been an amazing challenge working with this on this residency. Far more extraordinary and wondrous and challenging than I actually would have imagined it to be. I applied because I was reading Merlin Sheldrake's wonderful book, Entangled Lives, and actually the title, Your Body as a Planet, comes out of that idea. Comes out of his book. And reading that, I became more and more, he talks about this idea of fungi being this kaleidoscopic kingdom, which, because it's quite new in terms of research, it's one of the least studied kingdoms. The more you learn, the more you realize you know so little. And we were talking about that, Bridget, when we met you the other day. I have this feeling that I'm in the dark. And the more I'm hearing that it gets darker, but there are these little specks of light which are extraordinary and transfixing and make me want to learn more. And I think that is very much a place of art and science, of that understanding that there's so much to learn and being excited by that. And this idea of your body as a planet, that comes exactly from, so I'll read you the bit that comes from Entangled Lives. He says, for your community of microbes, your microbiome, your body is a planet. Some prefer the temperate forest of your scalp, some the arid plains of your forearm, and some the tropical forest of your crotch or armpit, your gut, your ears, your toes, your mouth, your eyes, your skin, and every surface, passage, and cavity you possess teem with bacteria and fungi. You carry around more microbes than your own cells. And I found that idea completely fascinating. And I've been spending the first part of the residency talking with the various scientists from the MRC Centre, and very much taking the place as this listener to learn these incredible stories. And this idea of I collated these, I've been collating these ideas with these cards. Here are some pictures of the cards. I didn't quite know how to, I wanted to be Tom Cruise actually in Minority Report. I don't know if anyone's seen that film where he has all this information in his investigation, but unfortunately I don't live in 2090, I'm in 2020. So I've resorted myself to the old fashioned cards. And I've been looking for ways in which the research inside the MRC Centre kind of intersects with my own practice and my interest. And when you asked me, Bridget, about that idea of the kind of microcosm, or this world through the microscope, the immediate resonance with my practice is that I've been working inside family a lot, and my family particularly. And I've always understood that work to be a sort of microcosm which expands out to global questions, that within the nuclei of the family, we can see a lot of the behaviors of human nature lies right there. And this idea of mycological thinking, like thinking through fungi, thinking through fungi about human behavior, really fits in with some of the people I've been reading over these past years: Ursula Le Guin, Octavia Butler. These are science speculative fiction writers, Donna Haraway, Bruno Latour, and then more recently Isabel Stengers, who is a philosopher of science. And I'm just beginning to read her work and find that very interesting. But this idea of a story, that the story creates another world, this idea that we can critique and invent alternative worlds and understand our ways of being in the world through the species we share and depend upon, feels very important. So the stories we tell ourselves, how can the story of fungi and this microbial story, this pathogenic story where actually fungi can kill us, help us re-embroider the stories that we tell ourselves and the place that we find ourselves right in within this biosphere that we are entirely dependent upon, that's created by organisms that we share this world, and we are dependent upon them for our own survival. And that fits in very well with someone like Donna Haraway, who says stories tell stories. It matters what systems systemize systems, and fungi, I think, can share a lot. And then going right back to the beginning of, say, man, where we see cave paintings, it really reminded me of John Berger, who took people down to see cave paintings, and he's an art historian, a wonderful art historian, and he used the phrase, in the beginning there was no fumbling. And this idea of going through the microscopic world into the world of fungi, I think of the artist Meredith Monk, who believes right now that there are two different important types of art. There's political art, but equally important are those that are thinking of what an alternative reality would be, to offer an antidote and go back to a more timeless concerns of existence on Earth. And I think that's about where do we go at this point as humans? And with the scientists standing on this entrance of one of the oldest labyrinths of life on Earth, fungi, I find that this idea of looking into this kind of deeply technology world through the microscope, it's this kind of challenge of how do I, as an artist who's interested in this timeless concerns, as I quote Meredith Monk, the artist, go through technology to explore that. And so I'm in the process of making a film where I'm working with the scientists, asking them for material that they've been gathering through their micro investigation through the microscopes, and looking at these extraordinary images. So I'm going to share you a few now. This is Candida albicans, which is ramifying a hyphae invading human oral tissue. And this idea that fungal hyphae is a sort of stream of embodiment rather than, say, a stream of consciousness. And that difference between humans and fungi perhaps being quite simple, that we put food into our bodies, but fungi put their bodies into the food. And I'm interested in that idea of how Barbara McClintock talks about when you're first encountering an organism, you need to get a feel for that organism, to develop a patience, to hear what the material has to say to you. And all of the scientists at the MRC have been so generously offering that chance for me to learn about and listen and learn how I might talk about it. So these are I'm sharing with you the types of images that I'm now collating from the scientists from their work without going into the specifics of what each one of these images is. This is the world that I've been seeing on a micro level. And the film will hopefully go from the micro into the macro, into the global, into the universal, maybe. That's the plan.
Brilliant, thank you. We're going through the images there.
Yeah. Do you want me to stop?
Okay, well, we'll just go over to Neil, perhaps. Yeah. And Neil, this idea of people moving into different worlds and talking about things that when we first start off talking about it, we don't know anything about. I know you're a real proponent and supporter of that kind of cross-disciplinary working. How do you think working, and we could say that we're all creativity is everywhere, but how do you find that crossover between working with artistic collaborators and research? Can you talk a bit more about how you think that works and what the benefits might be?
Yeah, absolutely. The first thing I would say is that I think some people might think, oh, there's a huge gulf, isn't there, between somebody who looks at the world with an artistic eye and one which dissects it with a scientific eye. But I would say that that is not quite the case when you think about how the world hits you. And I think scientists and artists definitely need to work together, because in the end, both are sort of looking at the world and they're trying to interpret the world. And they might use different tools, they might use different skills, but they're usually intrigued by, and possibly, not to put too fine a point, in awe of the beauty or even the ugliness of the world. And they want to try and express that, what that actually means to them. I really like the title that Léonie's given to her project, Your Body as a Planet, because I think that captures a lot. And he's talked about the wonderful book, The Entangled World, Entangled Life. And I think it's a very helpful metaphor, really, to think about your body as a planet. I mean, in the macroscopic world, we've just had G7 at Cornwall. Obviously, there's a huge increasing and rightful awareness that our world is one of great beauty, but also great fragility. And we need to look after it, and we're probably not doing that great a job of it. And our bodies, I suppose, are also pretty precious to us. We've only got one of them, and we've got to look after them as well. But like the planet Earth, we're not alone on our planet, and we're not the only species that counts. And as Léonie said, we share the body planet with many others. And in fact, on the planet Neil Gow, or the planet Léonie Hampton, or the planet Elaine Bignell, you've already heard there's at least as many cells of microbes as there are cells of our own types. And if you were to go into your gut, you would find that you'd have 100 or 1,000 different species in that area alone. And if you count the number of, say, genes which are on the planet you've got 100 times more foreign genes than you have human genes, different types of unique genes. So if an alien came down and started counting, what they see as a human being, they would have to assume that it is a kind of planet with many different citizens working together to achieve the outputs of what a body does. And in fact, remember also that every one of your cells, and every plant cell, every animal cell, inside those cells, there's an ancient bacterium. His job is to generate the energy that you need for me to talk to you and for you to walk away from the symposium, whatever you're doing. So I think these are all important elements. And after all, artists have always drawn, they've always painted and sculpted human bodies, for example, and nature. But the thing which Léonie's captured is that the thing about microbiology is that much of it is an invisible world. In fact, it wasn't even realized it was there until the 1670s, where the father figure of microbiology is considered to be Antony van Leeuwenhoek. And he was not a scientist. He was a member of the general public. He was somebody who made curtains and fabrics. He was a draper in the Netherlands. But he had a hobby of making lenses, and he made a lens sharp enough to see the first things which he called little animals, or he called them animalcules. And these were the images of microbes. And you can see back in there, the fungi are interesting because they span these different worlds. There are some fungi which we're very familiar with, but they're very beautiful. Some macroscopic fungi, mushrooms and things, but they're also the microscopic fungi. And so I think they're a fascinating group to look at. And keep in mind, there are about 5 times the number of fungal species than there are plant species. It's a huge, complex group of organisms. So there's a lot to be gained there. And when you look in the microscope, you can see a lot of aesthetic beauty. And actually, if you look beyond the level of the microscope and use things like X-rays to look at the proteins and molecular engines that drive these cells, they are also stunningly beautiful images. I just wanted to say one of the authors I really like reading because it's informative and hilarious at the same time is a physicist called Richard Feynman. And I think it was quite an interesting set of stories about art and science in his. So Feynman had a lifelong friend, a very close friend, who was an artist, whose name was Gerard Zothian, and they met in a pub, I believe, and they had an argument about a flower. And the argument sort of went that the the artist believed that the scientist was blinded to the aesthetic beauty of the flower because he wanted to pull it apart and understand its biochemistry and its molecular mechanisms. And the scientists completely disagreed with this perspective. And I've got a quote from what Feynman says. It says, "It's an appreciation of the mathematical beauty of nature, of how she works inside, a realization that the phenomenon that we see the result from, the complexity of the inner workings between atoms, a feeling of how dramatic and wonderful it is. It's of scientific awe." Awe is a great word, which I felt could be communicated through a drawing to someone who also had that emotion. In other words, they were on the same aesthetic plane. One saw it from a different angle, but actually they're both wanted to communicate about this. And these two men then agreed on consecutive Sundays to meet. And on one Sunday, the artist learnt about physics. And on the other Sunday, the physicist learned how to do, to perfect and to refine his drawing and his art. And they both got a huge lot out of it. And it's exactly like the project that Léonie is taking on with us. So I think when a scientist or an artist sees a fungus, and look at these pictures here, first of all, they're struck by the beauty of nature, in my view. And it's awestruck in many cases. But immediately, I think when you look at a picture like you've got in front of you now, also for a scientist, you start intuitively asking questions, right? Here, why are these all blue? What makes them blue? Is blue making an advantage for them? And why do we see blue in some parts of nature, not others? What does being blue mean? And these are aesthetic questions and scientific questions. What about the shape and the form? How do you get these different shapes? How does that encoded? If you're round when you're a microscopic cell, does that mean you're round when you're a bigger cell like a mushroom? And how do you get a big structure from simple cells with simple shapes? These are questions that an artist would ask just as well as a scientist. So I think that also I very much agree with the sentiment that you have to have a feel for the organism. And actually, I remember quite mischievously once, I was president of the British Mycological Society, and Paul Nurse, who some of you may have heard of, he's on the TV all the time. He's a brilliant molecular biologist, and he got the Nobel Prize many years ago, shared it for cancer. But actually, he actually only worked on fungi, he worked on yeast. And I presented him on behalf of the British Mycological Society a prize for his work. And it was a great honor to meet him. He's a terrific guy. And I knew it was going to be controversial because most of the British Mycological Society, or at least half of them, are people who collect mushrooms in nature. And they think that's proper mycology. And they didn't think that somebody like Paul Nurse, who wanted to dissect mechanisms all the time, would have that kind of aesthetic love for the organisms. But I knew these doubters would be wrong. And the first thing Paul said is, when he looked at his cells down the microscope, he could tell you on any one day whether that fungus was a happy fungus or a less happy fungus, because he knew how they thought and how they felt, and he had that feel for the organism. And I think any scientist who's worth his salt, who works on organisms, have to have that kind of feel. They have to almost think like that organism to understand what they actually do. And I really think that process can be helped by artists who might not be as blinded by mechanisms, but has always got the insight of understanding how it impacts on them and other people. So this is a great project that's going to really bring together all of these things at once.
Great. Okay. Thank you, Neil. That was really interesting. I'm sort of going to open it up to the three of you to kind of share some further ideas. Some of the things that we've talked about as well are around how, when you go into a different context, how do you decide what's important of all the things that are on offer to you that you could work on and explore? How do you prioritize what you focus on? I think something about mathematical modeling as well, because we haven't really touched on that, and how that's sort of maybe feeding into some of the thinking. And then finally, I think some of the information that's coming to light around how fungi communicate with each other and network with each other, I think that's really interesting. So out of everything that was offered, Léonie, how did you decide what to focus on in an artist residency?
In this particular artist residency, it's about what resonates. So it's going into a meeting with a scientist, understanding what they're studying, understanding their practice, and listening to what they want to share, what stories they tell, and certain things chime. Ooh, ooh, and writing those down, making the cards, doing a drawing, thinking through the stories that are being presented, and so really definitely not entering it with a preconceived idea of what I'm looking for, more entering into the space and finding what feels exciting.
May I just touch on one point? You've got 3 great questions, Bridget, but communication and fungi are fascinating. I think they are amongst the on the organisms on the planet Earth. They probably invented more communication systems than any other group of organisms. For example, if Léonie was to take a picture and to try and move her camera or to take up her paintbrush, she would only be able to do it because she's using electrical signals to communicate between her brain and her fingers and her hands. And actually, the fungi invented the electrical signal as well. And some of the most extraordinary electrical signals are ones which are from a fungus called the bootlace fungus, which is actually in the Guinness Book of Records is the biggest organism on the earth. Bootlace fungi, single colonies, are actually weigh more than blue whales, and they live in forests. And they're been genetically stable in some cases for more than 2,000 years. So in the Guinness Book of Records, down there's the oldest and biggest organisms. And being that big makes it very problematic to know what's happening at one end of your colony compared with another. And they have electrical signals, and they can say, I've got food over here, but not over there. So I'm going to break down all my filaments over there, remobilize their resources, and put them back over there where there's food to be had. And this is an extraordinary thing, but it's a primitive electrical signal which we use in the human brain. And certainly fungi communicate about each other. So if you were to look in your garden and find an ink cap fungus, which are very common in Devon and Cornwall, ink caps have multiple genders. So in human beings have only got males and females and other genders which are recognized, but that pales into insignificance compared with the ink cap, where there's more than 60,000 genders in that mushroom. And that fungus, what it has to do is to say, I will only interact and mate with a different genotype. And so they could discriminate each of the 64,000 different genotypes from one another and only fuse with ones which are dissimilar. So there are some amazingly sophisticated ways of communication which exist in the fungal world, which makes them endlessly fascinating to study.
Thank you. Yeah, it is absolutely fascinating.
So I could pick up on that briefly and just highlight that the way that fungi communicate with each other is incredible to study, and Neil's given us a great insight into that. Is incredibly similar to the way that human beings communicate with each other. And interestingly, we can use fungi to study that process in very fine detail. Essentially, it's an exchange of information that, in fungi, because they're simplistic organisms, is a ping pong mechanism of exchanging molecular or biophysical information. But it's immediate, and it's a ricochet of events that closely emulates the thought process of human beings when they interact and stimulate thought processes in each other. And it's a great simile for the conversations, whether they're verbal or exchanges of thought processes that have been important during our working together on this residency, and I can see that a question has come up in the chat about how we bridge the the linguistic barriers that are often built around different disciplines, and I spoke about the impact that this experience is having on all of us, actually, because I think that when we started out, it was quite a unilateral objective in terms of engaging and exciting communities. But well, maybe it wasn't a surprise to Léonie, but it was certainly a surprise to me that what could evolve from this was very great excitement about the way in which a fresh perspective on just an interesting problem and question could stimulate so much fruitful thought and commonality of approach, but fresh perspective that has altered the way that I think about the work that I do.
That's amazing. That's so interesting to hear. When you were talking about fungi earlier and how it works, and this idea of a simple organism, it was reminding me of that, what I was saying about John Berger talking about that sort of the no fumbling, the simplicity, and there's something perhaps in human behavior right now which can be too overcomplicated, not direct enough. And it reminds me of that Darwin said that if intelligence is based on how efficient a species becomes at doing the things they need to survive, and that in the crises we face right now makes me wonder, are we right to think we're the most intelligent species around? And we know that fungi has proved to be incredibly adaptive, resilient. It's been around since the beginning. So it's an interesting question, that idea of simplicity and intelligence, and yeah, what we can learn from this species.
It is, and you used a film analogy in your opening, Léonie, which was about Tom Cruise, I think, using this wonderful technology that I also wish we had. But I was reminded when you were talking about simplicity and the cave drawings about the movie Back to the Future. Okay, so and this microscope being a portal to look at ancient kingdoms of life that we couldn't understand until we developed the technology to gaze upon them and to begin studying them, right? So it just, identified another dimension, the fourth dimension, the one of time, and the rate at which we now have to study to catch up with those, millennia of missed observations, if you like.
So interesting.
Yeah. It's also humbling to remember that the fungi and humans are actually on the same kind of tree of life, the same branch of the tree in life. And in fact, fungi are closer relatives to animals than they are to plants, although people may think the opposite, but they're not. The plants branched off much earlier on. And even across those branches, there are things which are so similar that we're still sharing them to this day. And I think an example, going back to Paul Nurse, who got the Nobel Prize for cancer, was that the killer experiment he did was in his lab was to take a gene from a fungus, the fungus he worked on, the yeast, and to put it into a human cell and to show that fungal gene was able to drive cell division in a human cell, and in reverse, the human gene could compensate for the lack of the fungal gene. And that means that piece of the apparatus, which is fundamental for life itself and growth and cell division, which we all have to do, is so conserved that it's still able to, you can switch the component parts of the motor around and still drive that process, even though they diverged well over 1 billion years ago. So it's worth keeping in mind, how similar parts of life really are. And in fact, in fungi, which have been great models for disease, that a very large percentage, 25% of all the genes which go wrong and cause human diseases, you can study in fungal systems very simply. There are even genes for things like the gene for myotonic dystrophy, where we don't have muscles, of course. Well, we've got muscles, fungi don't have muscles, but the myotonic dystrophy gene is present in fungi, and you can study what it does in that. And many other genes which are important for human life can be studied in fungal cells, because we've got this shared history, and we're still, some of our biochemistry is still shared, and that's a massive problem for us in the MRC Centre, because it makes the finding of a true magic silver bullet, which just kills the fungus and not the organism in which it's growing, us, is quite challenging, because we're sharing so many common traits.
I was wondering, Neil, I remember when I met with you, you used an expression which really stayed with me. You said, we need to learn that we live on the shoulders of our microbial world, or of our microbes, and this idea that we could come tumbling down into the rubble of our own making. And I think with the pathogenic story, that if I were coming to this talk now, I'd want to know, well, what can I do as I leave this talk? What is it that I should be doing in my life, that you know, addresses what we're trying to talk about here? And I wonder what you might say to that.
Well, I think it's a very helpful element of society now that there's a renewed respect and concern about general biodiversity. Now, I sign up to make sure that, you know, tigers are well looked after, because I think it'd be a singular tragedy if we were to lose such a creature as the tiger. But at the same time, in the invisible world of microbiology and mycology, there are many fungi which we rely on in nature, which are potentially on the brink of extinction too. And it's actually much more difficult to study those, and because we don't know what they really are. We don't know all the fungi, we don't know all the microbes, but we do know that the things that we are doing to the planet, not just the human planet and the planet Earth, are a threat to that. And the biodiversity of fungi have given us a product for every single day of every life. You know, I bet everybody who's listening to this has used a fungal product today, tomorrow, and the rest of their lives. They may not even realize it is. You don't need to be, it's not just the obvious ones like bread and cheese and wine and beer, but if you had a wine gum today, you would have been eating citric acid, which came from aspergillus. And if you had a packet of cheese and onion crisps, you would have been using some yeast extract to flavor the cheese and onion crisps. There's no cheese or onion in cheese and onion crisps. And if you had an antibiotic because you had a strep throat, you might well have had a fungal antibiotic. And they have been a gold mine of products to support human existence. And we should be very mindful that the microbial world remains an incredibly rich source of things which we depend on. And so we need to respect the microbial world. It's much bigger than our macroscopic world. It will survive us and outlive us in the end, most likely. But in the meantime, we need to kind of understand how they work together.
Sorry, I dropped out for a bit there. And I don't know if this was covered earlier on, but one of the questions that came up in the chat was around navigating language, and it's back to communications. Léonie, when you go into this sort of context where it's very, it's a very kind of different environment, how do you kind of start off the conversations, and how do you, what techniques do you use to kind of bridge what may or may not be a gulf between yourself and the kind of context in which you're moving into? How do you approach that?
Well, I'm the first meeting I had with some of the team at the MRC, that question came up. They actually helped me with questions that I might want to ask, of which one of them was, you know, what is your team working on? Because within each scientist you see leading a team, there's a whole ecology behind them, and there's a whole team working to bring that research to us. So that was obviously an interesting question to ask when I first went to meet everyone. And I guess in the process of listening to what I was hearing, an awful lot I just didn't understand. So sometimes I would just allow that to wash over me, because it could become too stressful to try to imagine I could understand all of the language, the technical language that might be coming my way, and just the bits that, as I said, resonated, then just build on that. So something would make sense to me, and I'd ask a question back about that. So I think, and Lorna, who's been managing the residency, was also very helpful with that. She actually described that to me. Do allow things that are said just to wash over you. You know, your job is not there to have to understand it all. It's really, you're coming. I think that was a wonderful thing about the residency, is it's been set up as peer-to-peer. It's I come with my own skills and my own research and my own ways of understanding. And obviously, me being a visual artist, I did approach a lot of the very complex meetings just visually. They have these wonderful lab meetings on Fridays where teams from international parts of the world come together and they're showing their research to each other. And I've had the privilege of sitting in on some of them, and I'm in the dark. If I approach it visually, suddenly it becomes really interesting. And sometimes it's funny. They have these shapes that look like Pac-Man, or they have these graphs where I imagine like little animators coming along and detonating bombs, and just these things which mean nothing to me on a scientific level, but I can play with them. But I'm playing on a playful level visually, but I'm also playing in ways which are more meaningful about the sorts of things we've been talking about, where the images and the research they've been doing on a visual level is just extraordinary. And as Neil said, awestruck. I find myself awestruck an awful lot of the time.
And it is an amazing opportunity to be able to get access to all of this. How do you think in the longer term, what kind of fuel do you take from a residency such as this that might influence future work or ideas that you might develop further? What do you think are things you're going to take away from it?
Yeah, I think I applied for the residency purely out of being intrigued, and because I was reading Entangled Lives and was becoming more fascinated by fungi. And now I have become really, let's say, sort of entranced and inspired by the possibility of art and science working very closely together, because I do feel that if it weren't for some of the crises we face, I'd like to just be in a vegetable garden growing vegetables and reading books. And the reason I'm here is because I'm troubled by some of the crises that we face, and I want to do the best work I can as an artist. And I realize in the hands and with scientists, I can do so much more together. And particularly in the context of being in a pandemic, that could never feel truer right now. So much we are realizing so much of science that has brought us to having a vaccine so quickly. So we could say so much on that level, but it feels critically important at this juncture in time to listen to science and listen to scientists, and that we work in an interdisciplinary way on many levels.
Can I just ask, we'll move to the panel, the sort of audience questions in a minute. Can I just ask one final question though, which is around how a joint collaboration like this decides on what an outcome or an output could look like? Is it completely open-ended, or can expectations kind of fox the process sometimes? How do you kind of all get on the same page about an outcome?
I could come in.
I don't know who wants to answer that one.
I could start, Bridget, because actually I was involved. There were many dozens of applications for this residency, and the shortlist was actually quite long, and I was fortunate to be involved in that process that exposed me to a whole different world of disciplines, different techniques, materials, thought processes. And I learned as part of that process through working with Lorna and the initial stages of working with Léonie that it would be important not to place any constraints at all upon the process at the outset, and just to allow it to happen and to go where it would take us. And that was our decision from the outset. I think that was the position that we adopted, and it's been a remarkable process. And I would reiterate Leonie's point about it having opened up, you know, what most of the important discoveries are no-brainers, that were always there looking you in the face and you didn't appreciate. But it's incredibly important that this type of work becomes much more commonplace. This co-working becomes much more commonplace, and that we realise the importance of having these collaborations in place, and the impact that they can have on. It's very multidimensional.
I think that's part of the approach to arts and culture at the university, and the whole strategy around that at the university is to break down those barriers, and put people together that unfortunately previously haven't been able to do that, and it gives all those opportunities. So it's a great facility to have that, I think. So Laura and Helen, we're opening up to the audience questions. So I don't know if you want to sort of take the lead now.
Hey Bridget, we've had, I'm not sure if you can see the questions from before, but we had a question from Sarah Campbell for the whole panel on how language should be a bridge across arts and sciences, but we can actually end up in a ravine. How have you navigated this challenge for all the speakers, and what has worked for you when speaking across disciplines and knowledges?
Is that a question to all of us, to each of us?
Yes.
Shall I start? Well, you're making an assumption there, Sarah, that I have not felt in a ravine. I definitely have felt at times in a ravine of, there's so much information and it's all so extraordinary, and how do I choose between it? And there is a really different brain, I think, the creative, open, searching, exploring brain to the editing brain, and the one which goes yes to this, no to this, yes to this, no to this. And I think I'm only now stepping into that editing brain, but I definitely put myself in a ravine covered in a compost of stories and information until fairly recently. So I think that I'm now building that bridge through the film, through the creative process. And I'm so grateful to the team who decided to remain so open, that I applied with an idea which was primarily photographic as the outcome. But it was made very clear to me that that would not be necessary, that was the medium I ended up working with. And I think film is the right mode to hold the research, and so it's thanks to them being open to that I'm not bound by the idea of putting photographic output. And yeah, I think that my answer is that openness creates lots of ravines, and one goes into them, but one finds a way out. And together, I've not just done that on my own. There's been moments where each of the team in MRC have said things to me which have also sort of sent a little rope into my ravine and pulled me out.
One thing I just mention is this event, of course, is part of the Exchange Symposium, which is a celebration of the importance of public engagement. And of course, there is significant obstacles to language. All languages, create barriers to some extent, and it takes effort, sometimes to break through that. Science does create its own language. Some of our colleagues are immunologists. I'll tell you, Elaine will know what I mean. There's the language of immunology is a very complex one, for example. But actually, it's the same. If you're a specialist in any area, including university administration, Richard, they've got their own language. So do people who work in the finance sector. We all do it. And I think it's a very helpful process when you're doing interdisciplinary work to remember that, you know, the things that we're talking about can be overcomplicated by language, and it's incumbent on everybody to try and use a language which actually conveys a meaning without getting into technicalities or using unnecessary acronyms or specialist information. And I'm very pleased that virtually every grant that you write now within a, say, scientific or any other research purpose has got a lay summary. And the lay summary, I would tell you, is the most read part of any grant. And it's not just read by lay people, it's read first by the specialists too, because everybody's hungry for what the heck does this actually mean in plain language? You know, that is very important. And actually having conversations with Léonie and trying to get to really sometimes the deeper understanding of a piece of work, but trying to do it in a way which doesn't, you know, get immersed in technicalities is an incredibly useful skill, because if we're going to learn from each other, we have to communicate effectively with each other. And I think that language is important. I mean, Elaine does a lot of mathematical modeling, which is another challenge, but because mathematics has got massive things to offer. But it's a bit like speaking a foreign language to some. And Elaine, maybe you want to talk a bit about your experience, because you've had a long and successful relationship with a mathematical modeler. And that actually, to me, is a great example of, I understand not necessarily all the maths, but understand what the maths doing and what it means and why it's important. So, and that probably took quite a lot of effort to get that going.
There was, as with anything, there was an element of luck involved there, and I made the right connection with the right person at the right time, I think. But I see all barriers, obstacles, as tools to get fitter. And if you can think upon them in those terms, then they're good things, not bad things, and you can dissipate some of that frustration and overcome it. My foray into maths was an attempt to, well, it was first of all the realization that I couldn't possibly understand the complexity of what I was observing just with my brain or even a computer on its own, and that I needed to be able to use some different tools to understand how immune systems and pathogens interact. And the person I ended up working with was a bioengineer who had used maths to understand the flight of bumblebees. And so you can imagine that our first conversations were coming from entirely different angles, but in the end, it all boiled down to balance within systems, whether they're mechanical or biological, and how maths can describe that balance and the fundamental processes of feedback, you know, the physics of systems. And we persevered, and we together managed to establish a language that we both could understand, but that was simplistic enough that other people would understand it too. So you don't have to describe everything to understand the physics of a system. You just need to know what the dominant forces are and figure out how they interact. And just to finish on the maths thing, what's excellent about maths is that you don't have to be driven by preconceived ideas or accepted wisdom, because you can just change all of the variables all at once and see what happens that takes you by surprise. And it's a good alternative to using infection models that we have been forced to use in the past in our science.
Brilliant, thank you. I've got a final question that's come through from the chat, and there might be other ones, but this is, I think we've probably got time for one more, which is a question really for Léonie and possibly is a challenging one to end on. So Léonie, you suggested there are two modes of art which are important at present, political art and what the questioner are, but I think the question is, this is a political, is everything political? And do you think that really is influencing this project? Do you think it is the art that you're producing as a result, the piece you're producing as a result, will have an element of the political in it? Can you separate the two?
Yeah, that it's interesting. So I quoted Meredith Monk, who's an artist, and she, in the quote I use, she seems to separate them. At the time when I read that, I'm part of a collective, an artist collective called Still Moving, and we were working on a large light sculpture in Plymouth, a 67-meter sculpture that said "No New Worlds," a very political piece of art marking the sailing of the Mayflower. And I remember thinking that my goal as an artist would be to bring those two things together. I can't say whether I will achieve it, whether I do achieve it, but I think that's my ambition, that yes, it's together. It's hand in hand. You can't separate. For me, I don't want to separate those two ideas.
Brilliant. Okay, thank you. I think we've almost run out of time now. So this is just a quick wrap up to say thank you to the speakers and thank you to the audience as well for joining us. And do visit the Arts and Culture website at the university and also the Centre's website for more information, and hope you will see the project. That's the end of the session, and have a great afternoon, everybody, and thank you for joining us.